Hey guys, welcome to my YouTube channel. Today I'm going to discuss about the topic regarding the tissue culture. So what is the tissue culture and the process and applications of the tissue culture? So plant cell and how the tissue culture is cultured and get it further used for other applications like tissue culture main part is explant. So any part of the plant like leaf or apical meristem, embryo, cotyledons or hypocotyledon takes can be used as a for a process that is known as the that is known as the explant. Explant is nothing but any part of the plant like leaf, apical meristem or embryo, cotyledons acts as used for that material is known as explant. So these explants are transferred into nutrient media and in vitro is culture means inside the process. It doesn't go through it doesn't process outside the process. It comes under the like inside it happens. So it is regenerated through in vitro culture. It is observed in various research experiments and different plant organs of the different species response to in vitro culture conditions. So the response of the different plant organisms varies from in vitro culture. So any part of the plant like leaf may respond in vitro culture different response, and apical meristem responds differently in vitro culture. So for example, immature embryo are more responsive than apical meristem. Why? Because these apical meristems than immature embryo are more responsive than in vitro culture process, which are more responsive than leaf explant on particular tissue culture medium and culture condition. So plant regeneration in culture can be mostly achieved by morphogenesis. So morphogenesis is nothing but the external characters. Orthogenetic pathway, can be done by the two pathways, nothing but organogenesis and somatic embryogenesis. Organogenesis means the plant or the cell or a tissue in the plant tissue culture is known as the organs. So in a plant we cultured any part of the plant or tissue, it is known as organogenesis. Somatic embryogenesis is nothing but it is a vegetative propagation, means cutting a small part of the plant and placed in a culture, it grows into an entire plant is known as somatic. It is very closely related to parent cells. So coming to organogenesis, vegetative organs from cells or tissues in plant is known as the organogenesis. So firstly, the specialized cells of the explant start dividing under certain specific conditions from the mass. So this is the mass undifferentiated cell. This is the mass undifferentiated cell or part of it. This process is known as dedifferentiation. So here the process will go under the three process known as the dedifferentiation, redifferentiation and differentiation. So the cells of the explant start dividing under certain specific conditions forms the undifferentiated cell is known as the dedifferentiated process. This is the undifferentiated mass of the cells. It is followed by the organ called as primordia like shoot and root. So it undergoes the dedifferentiation process. First it undergoes the dedifferentiation process, then after redifferentiation process. Relative concentration of the growth hormones auxins plays an important role. So normally the plant cells or tissue cannot grow by their own. So some plant growth hormones are should need to be added, like auxins or cytokines, to help in the process of organogenesis. The process of formation of embryo from somatic cell is known as the somatic embryogenesis. Directly forming from the somatic cells, which means the vegetative part of the cells, it forms entirely the embryo, is known as somatic embryogenesis. Resulting embryos are called somatic embryos. Somatic embryogenesis follows the embryonic path of zygotic embryogenesis. Somatic embryos are very similar to zygotic except in the origin and size. These are very somatic embryos are very similar to zygotic embryos. They follow very similar in nature except in the size. The size is very big in nature and also the process of origin. Zygotic embryo has separate origin and somatic embryos has different origin. So here the some of the basic techniques of the plant tissue culture. So requirement for plant tissue culture. So here we follows the different steps in this. Like washing the area, the washing glass area plastic should be very neat in, and also it should be stored in a very dry and clean place. It should not enter any bacteria or fungal and various preparing various components of nutrient media for the preparation of nutrient. Again after that sterilization should be done, that at lower temperature, should not enter any other. Facilities for maintaining culture tissues in the aseptic condition under environment like and temperature. Temperature, humidity and lower temperature all are should be needed in for that culture media. Here the steps which included for the tissue culture. So there are a few steps for to be get followed for the plant tissue culture. First thing is nutrient media. Selecting the best and good nutrient media for the sterilization. And so for autoclaving is nothing but the sterilization. So while selecting the nutrient media and its sterilization, should not enter any microbial contamination, which means like bacteria, virus or fungi. So then after we'll go for the explant. What is meant by explant? Any part of the leaf or root or anything apical meristem is known as the explant. So any part or tissue organ is used in plant tissue culture generate differentiation cells, tissues or organs is known as the explant. So here most of the condition root or shoot apical meristems are and immature embryos are very easily grown. They are very easily used for the preparing of the explant than any other part of the like leaves or like that. Next, going through the surface sterilization of the explant. Means the surface should be get sterilized for the explant, should not contaminated from the microbial organism. So it should be done by disinfectants followed washing with the sterilized distilled water, and also used the high sodium hypochlorite. This is the most commonly used disinfectant for sterilization of the explant. Even though startingly we just sterilize it for the nutrient media, after that we'll taking explant, then after we'll get sterilized for the sterilization for the explant also. So explant harvested from seedling grown in sterilized condition need not to be sterilized again. So before only the explant is sterilized, here need not be get sterilized. So then after inoculation process should be takes place in a nutrient. Here we can see that small seeds will be get kept inside the glass, and then after it is placed some nutrient medium, then it start seedling process So here generating callus like structure in a fourth. Here we can see that in callus is formed with shoot buds from inoculated explant. Then after it start rootling media. Here we can say rootling media is observed. Then after it start producing the shoot, inoculated shoot rooting in. So then after it forms the plant rootlets, it transferred into the soil in the pot. This is the process of the plant tissue culture. Here we can see the tomato plant, how it grown through in vitro or plant tissue culture. So growing culture in this plant tissue culture under physical condition like temperature, humidity and soil, small calli are regenerated on cotyledons explant on suitable. Here we can see that small callus, undifferentiated mass of the tissue or the cell is known as the callus. So after this transfer of growing onto suitable media to generate roots, shoots and and other parts of the plant. So again it transferred into root after taking out from the nutrient it transferred into a soil. So then after hardening in the greenhouse or growth pot, it transferred into field conditions to plantlets. So plantlets are transferred into the soil for the growth of the plant. So this is the process of the tissue culture of the plant. First step, we are sterilized the nutrient medium. First step is sterilization, and next step is preparation of the explant. Preparation of the explant, and then after sterilization of the explant, then after inoculation. And then after inoculation it forms the small mass of the cell is known as callus. Undifferentiated mass of the cells or tissues is known as the callus is formed. Then after it forms the regenerating rooting media in the process. It slowly start producing the rooting in the inoculation shoot. Then after it forms the plantlet transfer to the soil in the process. This is the process of the preparation of the tissue culture. So if you like this video, please like, share and subscribe. Thank you for watching. In the next video, we will discuss about the applications of the plant tissue culture. Thank you for watching. Please keep supporting. Thank you.